So one of the reasons why I actually do this is because you're mastering genetics homework. It kind of goes a little bit of ahead. So I want to make sure that you guys are able to actually do these questions. Is everyone okay with me going ahead of lecture?
I mean, I'm fine with it, but
Yeah, that's fine.
Okay. Because I just want to make sure, because I had some students say that it won't be, it's if it gets confusing, I guess, like, if it gets confusing, be like, Nemi, stop. Tell me, tell me when you guys are lost, okay? Because I can't read minds, and it's even harder with videos off. So I won't be able to tell if you guys are confused or not. So if you guys are confused, stop me. Like, be like, Nemi, stop. I'm confused or something, okay? Okay, great. Let me just figure out something here because we have some technical difficulties with the student. Give me 1 second. Sorry about that. Okay, so the next thing we'll start off with presentations. So let me just stop my share. So, and where's this button? Okay. So who wants to go first?
I'll go.
Okay.
So I got the question, a female is diagnosed with the X-linked recessive gene disorder, hemophilia. In checking her medical history, the doctor will most likely discover that A, her father had hemophilia, her mother was normal. B, all siblings have hemophilia. C, there is no family history of hemophilia. And D, her mother has hemophilia and her father was normal. So I put A, and the reason why I chose A is because X-linked recessive genes are largely observed in males, mainly because they do not have an extra X, and females who do not develop this disorder are only known as heterogeneous carriers, and also this disorder tends to be lethal in females when it is homozygous, so the recessive disorder, and it's a sex-linked recessive disorder, so there has to be a family history of it. And so I showed this Punnett square to kind of like, give an overall of how this happens and how the father is giving her the recessive X link. And that's it.
Does that make sense?
Yeah.
Okay,
Yeah, that makes sense.
great. Who wants to go next? Guys. So I have Kelly and Ozzy. on for today. Guys,
Hello.
yes.
Yeah, I have my presentation too. Today.
Okay.
Do I share my? All right.
Yes, please.
All right. All right. So I had question two, which was a father with myotonic dystrophy has three daughters who all are carriers of mutant allele and two sons who are unaffected non-carriers. The three daughters have six sons, of which four are affected and two are not, and four daughters, of which two are carriers and two are not. From this description, what type of mutation is probably responsible for myotonic dystrophy? And so when I read that, I immediately thought that it was going to have something to do with the X chromosome due to the fact that it was passed down from the father to the daughters and they were carriers. So I decided to look into what X recessive inheritance was. And I found out that it is a mutation or disease that's passed down through a gene in the X chromosome, and it is presented only if it is homozygous recessive gene. And with that, I chose C, which is X-linked recessive. I said that because, you know, it passed down from the father to the daughters, and they were only carriers. If it was X-linked dominant, they would not be carriers, but they would have the disease themselves. And also the fact that their children had a 50-50 chance of getting the gene. Yeah.
Good. Does that make sense?
Yeehaw.
Yay.
Yeah.
Okay. Is Kelly in today? Okay. I guess Kelly's not in today, so, oh, wait, I have a question in the chat. Do we have to present on our day or can we present earlier? If there is a conflict, you can either by my office hours or if you want to go earlier during a session, you can. If you want to go in the following week, you can. You just got to let me know. Okay? Okay. So let's get back to our PowerPoint. Okay. So everyone can see the screen, right?
Yeah.
Okay. So the topic we're going to cover today is chromosome mapping and sex chromosome and determination. So before we go into chromosome mapping, let's talk about crossing over again. So what is crossing over? And how is it related to meiosis?
Isn't it like, all right, I'm gonna go. Isn't it like during metaphase where everything's in the middle and then like all of the chromosomes kind of like flip flop certain genes with each other?
It's actually between, it's in prophase, one, in
Okay.
meiosis, okay? So crossing over is essentially the exchange of genes between two homologous chromosomes. Essentially, they synapse and then they cross over during prophase one. Again, this allows variability to occur, and so another term for this is genetic recombination or recombination. And then genes that independently assort from each other, that ends up with high variability as well, okay? So one of the first topics that comes in when it comes to chromosome mapping is linkage. Linkage, so linked genes sit close together on a chromosome, making them likely to be inherited together. So in this case, since gene 1 and gene 2 over here are very close to each other, they are considered linked. Then, so these two genes are on the same chromosome, but not all genes on the same chromosome are linked, okay? So when there's so much distance between genes, there's more likely chance to have homologous recombination occur, or crossing over, right? So if you have another homologous chromosome, you're gonna see like crossing over happen like everywhere, like here, there, everywhere. Lord knows it happens every time, okay? So when that happens, then, okay, so that's one set that's not linked. And then when they're on two different chromosomes, they're not linked as well, okay? Okay. So meiotic consequences of linkage. So in this case, independent assortment, when it's independently assorting, there is no linkage. But because linked genes cannot assort themselves independently because they're together. They're like stuck together, okay? So linkage without crossing over, there's no crossing over, and there are only the parental genes, right? Whereas linkage with crossing over involves the generation of what is called recombinants. So essentially a recombinant is the mixture of these two guys, right? So it's like so we have the parents, which are homozygous here, I guess. Not homozygous. We have all dominant, all recessive. The parental ones will stay the same, but then the recombinant ones look different than the parent gametes, okay? So with complete linkage, it would be half of this guy would be, so if it's complete linkage, you will only see the parents. If there is no linkage, you'll see some recombination going on, okay? Does that make sense?
Yeah.
So, linkage affects expected ratios because it violates which of Mendel's postulates? The response in the chat. The chat lets me open it. Okay.
Is it D?
D?
That's what I think.
Yeah, it is. So again, linked genes cannot assort independently. And when we're talking about linkage, we're talking about multiple genes. So that kind of takes out A through C. And then you kind of have this one, right? So one of the next things that you can use linkage for is gene mapping. So it can be used to find the location of a gene by looking at how often different genes get inherited together. And that way you can actually use those genes and their distances between each other to and see where they're actually located, okay? So one of the things is that linked genes, again, linked genes will be inherited together more frequently. So when they are linked, recombination is less likely to occur between them. And if there is only less than 50% recombination, so that means the genes are linked. If it's anything over 50%, they will independently assort because they are not linked, okay? So this is how you would calculate the recombination frequency. It's the number of recombinant progeny over the total number, so it's the percentage of recombinants. So that's that. So gene mapping allows you to determine the order of the genes as well, and allows you to figure out the relative distances between the genes in what is called map units or centimorgans, depending on which context you're looking at. So in this case, we have recombination frequency, this guy, is also equal to the distance between genes. So 1 map unit is equal to 1% recombination frequency. Does that make sense?
Yeah.
Okay. So to calculate these guys, it's simple. So we have these progenies, right? So we have 49 of those guys with each of them recombinant, non-recombinant ones. And then there's one set that was had a crossover, a single crossover. And you see 2 non-recombinants and then 2 recombinants. So when you do that, you get, so again, as I said, number of recombinant progeny, so 1 plus 1 over the total number of progeny. So all of this added up times 100, which gives you 1% in this case. Does that make sense? Okay. So 2 genes that are 25 map units apart are expected to show blank. What do you guys think? What do we know?
B.
Yes. Oh, okay, so we know that anything under 50% is gonna have some kind of recombination showing. Oh no. So essentially it just says that it has 25% recombination because the distance itself tells you how much recombination it occurs. So essentially those guys are 25% recombination. So this is another question that is kind of overviewing everything that we just covered in a sense. Assume that a cross is made between big A, big B, little b, and little a, big little b, plants that are, and that the offspring occur in the following numbers. So you have 106 of this guy, 48 of this guy, 52 of this genotype, and then 94 of these guys. So these results are consistent with the following circumstance. Which one? So would you guys like me to walk you through this one? Okay. So what do we know? We know these are the parent genotypes, right? Because by the question, we know that these two are the parents. So these are parent ones. There's 106 of this guy, 94 of this guy. Then after figuring out the parental types, you know that these guys are the recombinant ones because you see that they are different from the parent ones. You have this many of these guys. Then what do? To figure out what's going on here, you need to figure out the recombination frequency or the distance between these guys. So in this case, the total number of recombinants is 100 when you add these two together. The total number of progeny of all of these guys together is 300. So the percent recombination ends up approximately 33% because you're putting it 100 over 300 times 100%, which gives you that. Then we know that 1% recombination is equal to 1 map unit. This means that 33% recombination is equal to 33 map units. Okay? So the answer to this guy will be D, because the linkage with approximately 33 map units between the two genes, gene loci. Does that make sense? Are we all cool? Because I know this is like a couple of questions from your homework too, so just want to make sure.
If we have another example, I'd like to go through it again.
Okay, I don't, oh, okay. So I do actually have one more. So 3 genes on Drosophila X chromosome are examined pairwise. It is determined that they have the following recombination rates. So yellow and white are 0.5%. White and miniature is 34.5%. Yellow and miniature are 35.5%. Which 2 genes are closest together? What do you guys think with this guy?
Is it A?
A.
Yes. It is A. So what do we know? We know that these are our recombination frequencies, the percent of recombinants in this system. We know that 1% recombination is equal to 1 map unit, right? So in this case, the distances between these genes are 0.5 map units, 34.5 map units, and 35.4 map units, right? So out of these guys, the lowest and the closest, since this is the lowest number, they are the closest. So this is the distance. So that means yellow and white are the closest, which makes A the answer. Does that make sense?
Yeah.
Did that kind of clarify the concept behind this?
Yeah, it clarifies the concept. I just got to practice the math.
It's essentially just taking the percentage of these guys. So when we go back to this slide, right? Oops. Sorry about that. I accidentally closed it off for you guys. Whoops. Okay. Can you guys see the screen?
Yeah.
Okay. So this case, so it's just adding up the number of recombinants, just making sure. So with this question, it's just narrow down what you're actually given. So just by looking at this without my explanation, what do we see here? We see that, okay, so we have 2 parent ones, right? Because we're seeing the same genotype for these guys. And then we have the 2 recombinant ones. So in this case, we know that these guys are the recombinant ones because they look different from these parent genotypes, right? So with this guy, that's essentially just narrowing it down, and then you take the percentage of the number of recombinants. So another way to practice these questions is just changing up the numbers, what you would get, right? Does that make sense?
Yeah.
Okay. I hope that clarifies the stuff behind this. So we covered this guy. Are we cool with this question?
Yeah.
So the next thing you can do with chromosome mapping is actually mapping the order of these genes. So this question, I'm going to walk you guys through it. And then the next question, I'll let you guys kind of talk through it with me, okay? So a trihybrid cross produces the following recombination frequency. So between V and CV, we have 6.4 map units. In this one, we have V and CT, which is 13.2 map units. And then CV and CT, we have 19.6 map units, right? So what is their order on the chromosome? So one of the first things you would do in a question like this is put down which is the longest map unit or recombination frequency, right? Because just by looking at this, you know that these two are the furthest apart from each other. Then you would go down to the next highest one, and then go down that list of map units. So in this case, we know that V and CV have a shorter distance between each other. So it's, and then we have 13.2 for V and CT. That's saying that there is some distance. So it's telling you that there's like, so with V, there could be 3 possible choices, right? One could be here, one could be here, one could be here. But then when you look at these distances, this number is not higher than this guy. So we're gonna see automatically that it would end up in the middle. Does that make sense? So the answer to this question would be A. Does that make sense? Okay. So this next question, recombination frequencies between 4 loci in
No, Jane.
in corn are shown in the following table. So the loci here, we have these different loci and the recombination frequencies. What are the order of these genes? What do you guys think? Do you guys want me to go over it together?
Yeah.
Yes, please.
Okay. So what is the first thing we do?
Well, do you go with the longest the chain?
Yes. So the longest recombination frequency here we see is 55, right? So that means R and L are very far apart, right? So we have the 55 map units for R and L. We have that written out. Then we go to the next highest, which is involving Q, right? Q has 40 map units away from L. Let's see where it lies around R. So Q and R are 15 apart. So in this case, you would know that it is in between them because they actually add up to 55, right? 40 plus 50, 15 equals 55. So there is some distance. So, because it wouldn't be here because it's not higher than this guy. And it's not over here because the other one's not higher. So in this case, it would just end up with Q being closer to R, but farther away from L. Then we look at the next remaining one, right? So we have W. W is 20 map units away from Q and L, right? So that means with Q and L, you already have it at 40. So when you look at these numbers, you can tell that it is smack in the middle of Q and L, right? Does that make sense? So that's how you would get this order. Does that make sense?
Yeah, thank you.
Okay. Everyone good with this kind of questions?
Yeah.
So I know I am a lot ahead with lecture at this point, but I want to cover this as well, just because you guys have an exam coming up in 2 weeks, I think. So I want to make sure that we cover enough so that you guys can keep with that. So the next topic we're going to cover is sex determination and sex chromosomes. So one of the first things we look into is the sex chromosome compositions themselves. So in this case, so obviously we have some X, 2 X's and 2 X and a Y for like humans. This is the same for Drosophila and mice. So in this case, this setup is called ligase mode. And so for C. elegans, this guy here, the little cute little nematodes, these guys have an XX or an XO. So they don't have a second, I guess, sex chromosome. So it denotes, for males, it's denoted as XO. This mode is called protonor mode. So I guess like I've seen in previous years that Dr. Usuna asked questions about like which mode is an example of protonor mode. When you think protonor mode, think of C. elegans. So this is actually the lab I work in. We are the only C. elegans lab, and this here, we're gonna call her Lena. Lena is, okay, so this is just, I guess, Lena the C. elegans likes it when people get this question right. It's an odd little nematode, you know. So remember this guy, please, or we're gonna have an unhappy Lena. So please do remember it. Okay. So any questions about these different modes? Okay. So what are the main components of the Y chromosome? Okay, so there are three parts that are crucial for the Y chromosome. The first one is pseudo-autosomal region, sex determining region of the Y, or SRY. And then you have the male specific region of the Y chromosome. This specific part does not recombine whatsoever. This, on the other hand, has homology. PAR has homology with the X chromosome. So that's where, that's the region where it's important in synapses. And then you have the sex determining region of the Y, which encodes the protein called TDF, or testes determining factor. So this is a picture of the Y, depiction of the Y chromosome. So you have the PAR regions at the ends, and the male specific region, which includes the SRY. So this area here doesn't recombine, but since PAR has a homology with the X chromosome, that's how the X chromosome and the Y chromosome end up aligning as homologous chromosomes. Does that make sense? Okay. So what do the studies of chromosomal abnormalities tell you about Y chromosomes in humans? So we have a bunch of scenarios here, right? So we have XX, which are females, X, which is X with no other chromosome. That would be Turner syndrome in females, but they're still females. Then you have three X's and they're still males, females. Then we have this guy, X and a Y, that's our normal male. Then we have XXY, which are still males too, even though we have an extra chromosome, which is a syndrome called Klinefelter. And then with males, you have XYY as well. So this is like a scenario where they looked at all these different individuals with different genotypes of these X and Y chromosomes. What does this tell you guys about the Y chromosome? It determines maleness, right? Because no matter how many X's, you still ended up with female. But then one Y chromosome came in, you're a male, right? Does this make sense?
Yeah.
Yeah.
Okay, good. So what is a bar body? Where is it found in the cell? So bar bodies are also called sex chromatin bodies. They are essentially darkly stained bodies in the female interphase cells. And essentially a bar body is an inactive X chromosome, okay? And they are highly condensed, so you cannot reach it for gene transcription or all that fun stuff of gene expression, right? So this is a bar body. You can see it's like tightly condensed DNA itself. So one of the things to notice is that only one, if there is more than one X chromosome, only one remains active. So what do you guys think with Klinefelter syndrome? How many bar bodies would you see?
2?
You have one chromosome remains active, right? So bar bodies are inactive X chromosomes.
Is it one?
Yes. Does that make sense? What about this
Kind
guy?
This is
Oh.
0.
Yep, this is 0 because if you have one X, that X remains active.
So if you have X and Y, both of them will neither of them will be bar bodies. But if you have two X's, like a female would have two X's, one of them would be a bar body.
Mm-hmm.
Okay.
Yep. So if ignore the Y here, if you had just two X's, it would just be one bar body because you have one other X to be active, right? In this case, there wouldn't be any bar bodies because you only have one X, and that one X has to remain active for it to be viable, right?
I have a question. Sorry. For the Klinefelter syndrome, does the bar body, like, is it just like a random chance for it to cancel out the X or the Y? Because like both work if you cancel it, or is it just for X's?
So bar bodies are specifically showing inactive X chromosomes. For Klinefelter, it cannot actually cancel out any of the issues going on because it's not complete inactivation. And one of the other questions I have will actually answer that question too.
Okay.
So just bear with me.
All right.
But so what do you guys think about this one? How many bar bodies here?
Three.
What does the 47 mean?
So 47 just means that there's one more chromosome that is added on to it. So it's essentially saying that it's varied from the regular 46 chromosomes that's present.
Oh, so is it also one?
So 2 are bar bodies?
2. So in this case, the 47 is the syndrome name, I guess, because it's adding on one more chromosome here. And in this case specifically, the genotype is telling you that there is an extra X chromosome. So in this case, one remains active, and then you end up with 2 inactive ones, which end up inactive. Does that make sense?
Yeah, it makes sense.
So what about this guy?
So then 3 of them are inactive.
Yep, that is correct. So that is, okay. What about this guy? The 47 is just saying it has an extra chromosome. Remember that it's only X chromosomes that are getting inactive, not Y chromosomes.
So none of them are inactive.
Yep, so it's 0. Does that make sense?
Yeah.
Okay. So the mechanism of inactivation. So essentially there is an X inactivation center that's called XIC. It's essentially where it's on the X chromosomes itself where it pairs. It's only active on the X chromosome that's inactivated. So the X inactivation center is activated on the X chromosome that is inactivated. So in this case, it contains several genes important for inactivation. 3 non-coding RNAs play critical roles in this process. These include the cis gene or X inactive specific transcript. This is critical for inactivation. If you have a deletion in this guy, then you'll see chromosome inactivation. So if this gets deleted, there is no inactivation going on. The RNA itself carries the gene that produces it. Okay. Then you have a gene called T6. This transcribes a non-coding antisense RNA partner. So essentially, this is the complement to this guy, except the antisense side. Then you have excite gene, which is transcribing a non-coding RNA. So essentially, the XIC, that center, it produces all of these RNAs, and they're like, oh, okay, we're gonna go to each of these different genes and block it from being transcribed. So that is essentially how X inactivation works. Now there is something called the Lion hypothesis. And this states that inactivation of these X chromosomes are random. And it occurs at an early stage of embryonic development called the blastocyst stage. I can't talk today. All descendant cells have the same X inactivation. So an example of this are calico cats or tortoise shell cats and their fur pattern. The mosaic pattern actually comes from the patches of gene expression, depending on which X chromosome is inactive. The reason why this occurs is that 15% of the genes actually escape that because it's happening at the blastocyst stage and not earlier than that. So that's how you end up with the mosaic pattern. So like some of them are still escaping that inactivation. So you see that mosaic pattern. Does that make sense? Yes, no, maybe so.
Kinda.
Where am I losing you guys? So essentially, the Lion hypothesis is just saying that it's random. And it starts at the stage of blastocyst. So essentially, this, like, it's, even though it says it's completely, it's not exactly completely inactivated because in the beginning of development, you see some inactivation, some genes that escape the inactivation process, right? So that's how you end up with two different colors on a cat, because this is saying that it's incomplete inactivation.
That makes more sense.
Okay, good. Any other questions, concerns about this slide? And this only occurs in females. Why? Why do they think that it is? Females are the only ones with two X chromosomes, right? So this mosaic pattern would end up with a random inactivation part. Males don't show it because they only express one of the potential X-linked alleles. That's why you don't see it in males as often as you would see it in females. Does that make sense?
My sister has a male calico.
Ooh.
Does that mean he has like two X chromosomes and a Y?
I think it might have to do with the PAR region, so the pseudo-autosomal region. It does have homology to the X chromosome, so it might have to do with that, but I'm not entirely sure. That could be a possible reason. You would have to genotype your cat or to figure that out. But does that make sense?
Yeah, that makes sense.
So why does inactivation not prevent these syndromes, such as Klinefelter or triple X? I think that comes back to Mario's question, right?
I don't even
So-
remember what my question was anymore.
Okay, so I think it was more like if it would cancel out with the X chromosomes, which it does not actually cancel it out because it would still be male in this case for this guy because you still have the Y chromosome. The Y chromosome automatically tells you that it's a male. But in this case, why X inactivation kind of prevents the syndrome is it doesn't prevent it because inactivation doesn't occur until the blastocyst stage. So there is approximately this many stages before you get to the blastocyst stage. This means that the inactivation itself is not entirely complete. And this is what I said earlier with the whole 15% of genes escaping, because since it's only starting at this stage, they could already be expressing things already within this area itself, but and essentially escape that inactivation process. So essentially, this comes to a point where the balance of the gene expression from the X chromosome can still be altered in these syndromes. Does that make sense?
Yeah.
So that is what I have for you guys. So for attendance today, I am going to have you guys do the whole type here in the chat. So do you guys have any questions?
No.
Concerns?
No.
Oh, thank you.
Everyone happy?
Yeah.
Okay,
Yeah.
so I'm gonna give you, oh, hold on, don't run away. I just have to tell you guys one thing. So the quiz will be based on whatever I covered with chromosome mapping. So make sure to look at that, okay?
All right.
Thank you. Have a good night.
You too.
Thank you.
Thanks. Good night.
Good night. Did everyone sign in? Or yeah,
Good
okay,
night.
great.